Okay, everyone. Welcome to part 2B, chemical and biological synthesis of peptides. In our first section, we looked at the energetics of peptide bond synthesis. So we've completed that. So we're going to talk about how biology makes peptides. All right. So how our bodies, the bodies of prokaryotes, other eukaryotic species. Then after that, later on, we'll talk about chemical synthesis of peptides. All right. You understand that we need to activate our amino acids in order for them to couple. So we talked about that in the first part, and you've also learned all these different 20 amino acids. Now, if you were to put them together and activate all of them and try to make a peptide, you know that it would just be complete chaos because how can you get the right amino acids at the right place at the right time? Right? So we have to do this selectively. How do we control the reaction selectively and in the right sequence and at room temperature in vivo? So biological systems use something called an activation strategy, and synthetic systems, chemistry uses a protection and activation strategy. Now, I'm going to talk about protecting groups in the chemical synthesis unit, but I just want to emphasize that protecting groups are really used to take and mask undesired nucleophiles from cross-reacting. So for example, in this reaction in which we have a serine molecule, and if these two serines were to come together, we know that this amine from the serine would need to react at the carboxylic portion of another serine. However, we need to take into account the high functional groups like thiols, for example, or amines from lysines, for example, need to be protected. Now, however, biological activation methods, such as a ribosome, actually, they doesn't require protection. Okay, so it actually makes things actually a lot easier. Okay, here's a image that you have seen before, maybe not this exact image, but you certainly have heard of the ribosome, which is, like I said, this macromolecular complex that facilitates protein synthesis. And really what it does is, and how it works, is really quite elegant and amazing. So embedded within the central unit of the ribosome, you can see here we have a messenger RNA. And the messenger RNA, you can think of it as a coded thread that passes through. And every 3 nucleotides, there's a specific codon and a specific code that it corresponds to. And then what brings in the correct amino acids for that code is something called a transfer RNA, tRNA, transfer RNA, that codes for a particular amino acid. And this will now hydrogen bond and bring the correct amino acid at the right spot. So you can see here you have a growing polypeptide chain. This mRNA feeds through, and then it looks like the next amino acid is glycine. And then you get amide bond formation. It shuttles over, and then you get the next incoming amino acid. So it's actually really, really fast and efficient process. And it uses an activation strategy, just like we learned in the last unit, to make proteins on the ribosome. Okay, so what is the activation strategy? So it uses an ester to activate the acid. All right, so here is an example of a formulated methionine. So the formyl group is naturally protected at the first amino acid site. Probably just so it doesn't react with other functional groups. Now, in order for this to, so this is our desired electrophile where we want the amide bond to start being made. In order for this to be intercepted by a nucleophile, we need to first activate it. Recall, this is a lousy electrophile because you get strong resonance delocalization. So what this molecule is going to do first, it's actually going to react with this molecule, a very well-known molecule. So I'm just going to take some time drawing it. We have a number of different oxygen, phosphorus bonds here. Here we have a sugar, and then attached to the sugar we have a heterocyclic base. Okay, so we have a triphosphate, and then this is an adenosine molecule. So you have seen this before. This is actually called adenosine triphosphate. So I know with like glycolysis and other energy pathways, you've used and seen this molecule before. So adenosine triphosphate is also used to help load and activate this amino acid. So how it works is you have a number of different electrophilic phosphorus positions here, alpha, beta, gamma. What it will do is it will react specifically at the alpha phosphorus. Again, this is governed by localization of this amino acid with the particular enzyme to get this reaction to occur. And what you will get is your methionine with now not a carboxylate, but a phosphate ester group attached. And recall that phosphate esters were one of those types of functional groups that were reactive enough, but not too reactive and not too inert. So we classify one of these functional groups as being, just right in terms of using that Goldilocks analogy. Okay, so we've activated this thing. What you will release off is diphosphate as a byproduct. We don't really need to worry about that too much. But what we need to do now is we need to take this amino acid, and if you think back to the previous slide, we have to now get this to interact with something called a transfer RNA. So transfer RNA is a very large macromolecular complex. It's an RNA molecule that is around 75 to 90 nucleotides in length. So there's lots of different functionalities within it. But what I'm going to do is I'm just going to abbreviate transfer RNA as tRNA with a hydroxyl group. Okay, and I'll give you some more insight in terms of which hydroxyl group that is on in the future slides. So what this transfer RNA does, this amino acid that we've generated here has to now transfer to the transfer RNA so it can get to the ribosome. So we now have here, right, this is our active phosphate ester. So now the transfer RNA with a hydroxyl group can come, and it can easily come up, electrons go up, and I'm just going to short form this. Electrons come up, then they come back down, and then you break that carbon-oxygen bond. So the net effect is that you have this formal methionine group attached to a transfer RNA. And then the transfer RNA contains that code where it can actually meet at a particular site on the ribosome. Okay. And you can see that your byproduct here, if this is ATP, your byproduct here will be AMP, adenosine monophosphate. All right, so let's take a closer look at the tRNA and how it gets activated by something called a Trna Synthetase. All right, so this is, as I mentioned, tRNA. So you can see it's a rather large looking molecule. This is a three-dimensional structure of it actually getting very close contacts with this large enzyme here in gray, which is called a tRNA synthetase. So you can imagine there's many different tRNAs out there. So you have to get the right amino acid attached to the right tRNA. Okay, so there are, depending on the sequence, it could be slight changes in sequence here that help govern the specificity to the outer surface of the protein. And then embedded here, there's an activation site, which is, there's a hydrophobic pocket for the R chains of the different amino acids. And so you can have very small amino acids like alanine, or very large ones like tyrosine or tryptophan, which are aromatic. And depending on how they fit into that active site will determine which tRNA goes to which amino acid. It's also very stereo-specific, meaning that if you have, as you know, we use L amino acids, but if you try using an R amino acid, the stereochemistry will not fit in the right orientation. Okay, so this is your activation site, which contains a hydrophobic or an hydro pocket for the R chains of amino acids. And as you know, there are around 20 natural amino acids. What we will talk about later, though, is trying to maximize or saying, well, could we use other non-natural amino acids that could fit in here to maybe expand the scope of the protein machinery? Okay, we'll talk about that later. Start talking about unnatural amino acid synthesis. And then down here is the anticodon loop, where it binds to not only the Trna synthetase, but also has to bind to the messenger RNA. All right, so let's take a look at a specific example here. This is a tyrosine, or what we call tyrosyl Trna synthase from tyrosine. All right, so here's tyrosine. There are actually 3 potential nucleophiles, okay? L and enantiomer only. Here's a potential nucleophile. The NH3 group, once it's deprotonated, becomes NH2. That's also a potential nucleophile. So what happens, like we'd seen previously, is that this will bind and react with ATP, adenosine triphosphate. Okay, so we've gone through this mechanism. It's also specific too, right? There's actually 3 potential electrophiles, 3 potential reactive phosphorus. Right, you have the alpha, beta, and gamma. It just reacts at the alpha because how it fits within the active site of the Trna synthase. Then you generate this molecule here, like we've shown on the previous slide. And then here, this molecule here now binds with tRNA and only the tRNA tyrosine only. And it's a 3' hydroxyl group that will react, okay? So this will come and this will react here and kick off adenosine monophosphate. Okay, so this is your phosphate ester. And then now what we've done is we've gone from a carboxylate, which is inactive, to now we've created an ester, okay? And this ester is much higher in energy compared to a carboxylate. So how are we going to make our amino acids? This is going to be our reactive functional group that's going to be used over and over again in order to make amide bonds. So in this slide here, you can see we have our first amino acid with tRNA attached to it. The second amino acid, okay, so let's call this tRNA1, and let's call this tRNA2, which now has a second amino acid. So now, because we have an activated ester, the amine group can come react at this carbonyl, electrons go up, they come back down, and then it kicks off the tRNA. So the net effect is you've now created this amide bond. It shifts on the ribosome, and now you get your third amino acid coming in. Again, this third amino acid will have a tRNA attached to it. We'll call that tRNA3, and it'll go through the same type of mechanism that we've seen in the previous one, okay? And then amino acid 4 comes in, so on and so forth. So the desired polypeptide is made. At the end of it, at the last sequence, where you have that last tRNA attached, you simply need to hydrolyze that to release off your free polypeptide, okay? And again, here's a 2-dimensional drawing showing the messenger RNA, showing complementarity with the anticodon and the codon loop, and then the 3' hydroxyl here, showing the actual ester linkage here that is used for activation to accept the incoming nucleophile. So that's really what it is. It's really quite simple and elegant, and doesn't use any protection schemes because biology just brings the correct nucleophile to the correct electrophile. So what does biology use? In summary, it uses an activation strategy to make a phosphate ester. Specific synthetases, specific transfer RNA synthetases are unique for each amino acid, and they position the nucleophilic carboxylate to the appropriate electrophilic phosphoanhydrate of ATP. Okay, and this occurs really rapidly. You can make proteins and peptides and ribosomes depending on the sequence and which codon you're using, but you're looking at thousands of amino acids per second, so it's extremely efficient. And however, there are limitations to this, and we're going to discuss chemical synthesis and how we can actually make peptides using chemical synthesis in the next part. Okay, thank you very much.